So I'm Bruce Leuchter, the CEO of Neurvati Neurosciences and GRIN Therapeutics. I'm a neurologist and a psychiatrist by training. I practiced neuropsychopharmacology for about 10 years all in, and then spent a number of years in various different roles on Wall Street, equity research, investment banking, M&A, and have now been at the helm here at Neurvati and GRIN since September of 21. And we are focused on a rare pediatric epilepsy called GRIN-related neurodevelopmental disorder with an orally bioavailable small molecule called radiperdil that is a negative allosteric modulator at the NR2B receptor. And we've led with GRIN-related neurodevelopmental disorder because it's a genetically defined disease that leads to hyperactivity at the NMDA receptor, which is where we bind with our experimental therapeutic, radiperdil. But we also knew that the NR2B biology is relevant in diseases beyond GRIN-related NDD, which is what led us to pursue a basket study, phase 1b basket study, in two additional rare pediatric epilepsies called tuberous sclerosis complex and focal cortical dysplasia. Both of those disorders are impacted by NR2B biology. And now we've advanced into that study, are enrolling patients, and hope to be able to produce data, proof of concept data in 2025. These are interesting disorders insofar as you can identify them on an MRI scan of the brain, which is not typical for a lot of the epilepsies that we ordinarily think about. So these are patients who present typically as children with either epilepsy or developmental delay or a combination of both. And they go through a standard workup by the physician, pediatric neurologist, or potentially a pediatrician, but typically a pediatric neurologist, epileptologist. And as they evaluate the patient, they ultimately go to MRI scan of the brain. And when the MRI scan of the brain comes back, you see what they call hyperintense lesions. That's technical terminology for understanding neuroimaging. And to make it a bit simpler, these are bright lesions in the brain where the seizures initiate. And a lot of these patients are treated, most, if not all of these patients are treated empirically with broad spectrum antiepileptics. More than half of them are treatment resistant to those broad spectrum antiepileptics, which are not disease specific, so are good agents to use in patients who've got epilepsy, but there's nothing about those approved therapies that are specific to TSC and FCD. So our view is that once we're able to work these patients up and determine that they are treatment resistant to approved therapies, we're able to bring them into the ongoing study and have them titrated on radiperdil, our experimental therapeutic, which targets the NR2B receptor. And in these patients, they have a hyperexpression of NR2B in the lesions in the brain, which is not surprising per se, because underdeveloped brain has an overexpression of NR2B. So some of the work that's been done around these disorders when children or young adults go to surgery, because that's what happens in some of these cases, they are treatment resistant to approved antiepileptics, and they are evaluated by epilepsy surgeons who are then, in certain cases, able to go in and resect the tissue that is abnormal. And when we evaluate that tissue, we see a hyperexpression of the NR2B receptor. So we are using our NR2B negative allosteric modulator to treat these patients in a disease-specific way. But they don't only suffer from seizures, much like we think about brain-related neurodevelopmental disorder, where seizures may be a primary focus. There are all sorts of other symptoms that these patients experience, and that's true also for TSC and FCD. So our goal is to be able to have an impact on the broader syndrome. The Astroscape trial is a 1B study, open label 1B study. It is enrolling 30 patients, both with TSC and FCD. It's what we call a basket study, where you can include more than one disorder in the study. We've done that in this case. So a total of 30 patients, open label. Those patients will be screened for a 4-week period. The purpose of doing that is to establish a baseline seizure count, because the way we assess efficacy of this drug in clinical trials is to determine how much decrease in seizure frequency these patients can experience when they're on our experimental therapeutic, radiprodil. So we study them, observe them for 4 weeks, 28 days, and we count the number of motor seizures that they have. And there are ways of adjudicating that with the Epilepsy Consortium and others, such that we are counting the right kinds of seizures. Patients will remain on their antiepileptics when they enroll into the study, so we're not making changes to their baseline therapy, but we're observing them for 28 days to see what their baseline seizure count is. Once we get through that 4-week period, we roll them forward into the titration period of the study, which in this particular study is about 4 weeks long. And we'll be titrating the dose upward so that patients can receive the necessary amount of medicine. And then we count for another period of time. This is called the maintenance period. It's the 12-week period. And we count for that 12-week period how many seizures they have. And then we measure the number of seizures they've had during the maintenance period and compare that to the number of seizures they had in the observational period. And that'll be the primary outcome. Well, the primary outcome measure is in fact safety and tolerability, but as it relates to seizure count, that will be how we assess patients in this study. And of course, there'll be an open label extension period, as there are in most studies, to keep patients on drug once that 12-week period has completed. Well, we are enrolling patients right now, and our goal is that over the coming months, we'll have complete enrollment in the study. We'll have patients progress through the totality of the study through the 12-week maintenance period, and then be able to report proof of concept data somewhere in the middle of 2025. Targeted therapies are critically important really across all therapeutic areas, certainly in neuroscience, including both neurology and psychiatry, where there's been a very big push to develop drugs that have a targeted effect on the underlying disorder. The view there is that if you can treat the symptoms and you can treat the underlying biology associated with the disorder, you can potentially have more clinical benefit. And that's what we've done in all of our studies. We've done it for GRIN-related neurodevelopmental disorder, and we're doing it as well for TSC and FCD. Now, that's not meant to take anything away from the significant clinical impact that broad spectrum agents have. And there are many of those drugs that have been approved, many that are currently in development. They play a critical role in treating patients because they can quite impressively manage the symptoms. But the difference associated with those types of therapies versus targeted therapies is that they don't necessarily target the underlying biology associated with the disease. And that's what we're doing with radiprodil as a negative allosteric modulator at the NR2B subtype receptor, where we can identify disorders or diseases that are driven in total or in part by dysfunctional NMDA neural circuitry or NR2B neural circuitry. And by doing that, we can not just treat the symptoms, but we can also treat the underlying disorder. And that would be a big change in how we think about treating epilepsies. There are companies, our peers in the industry, that are aiming to do similar things. And to your question, we definitely see this as a movement in neuroscience drug development where companies are quite focused on being able to manage the underlying disease process as opposed to just managing the symptoms. Both are important, to be sure, and patients benefit from both. But if you can accomplish the latter, which is targeting the underlying biology, you can potentially have greater therapeutic benefit on patients.